Actually, so on the depletion, actually, we have experiments with quite higher depletion. Normally, the average depletion rate with log depletion is about 4. Here we have reached actually 4.75. And to get just an impression, what does it mean in different publications? This is mixed actually between non-malignant, not malignant. You see here also that we have on the TC alpha beta side actually we are averaged, or we are better than average actually in the log depletion. The graft composition. So we managed to have a high amount of the CD34 cells, and we really managed also to get as low as possible alpha beta cells. So we really reduce highly the number of alpha beta cells of the alloreactive sites, also for the CD20 sites to avoid PDLD. But we have also in this graft actually a high number of NK cells and of gamma delta cells. Gamma delta cells. We have got a good number here. So it was quite a successful graft we found here, and we have also just only, I'm not going through all the data, just only to confirm, we have seen this also in different papers that the outcome of the graft composition looks really quite nice in our scenario. Then engraftment rate, so we talked about now about really the quick engraftment, and we could confirm that's quite well. So for the neutrophils we have an average median time of 12 days, for the platelets we have 15 days actually median time, and of course what I said, because due to the ATG we have a final engraftment of 29 to 30 patients. And you can see here nicely actually the engraftment works out really fast. So the immune reconstitution, I have summarized the picture here of 14 days and 100 days. So you can see the CD3 in all compartments, actually CD4 and CD8 cells really come up really good, really fast actually. But NK cells, they start as day 14, they got a peak, they drop down and got a plateau. I don't know actually why NK really get a peak and then lower it down. It's not. I asked many physicians, maybe you have an answer for me, why NK cells peak and then they drop down and get a plateau instead of just peak and get a plateau. I have really no explanation for this. But we see also a really nice immune reconstitution here on day 100 on the CD19 cells. So now it's getting interesting, how it looks really in acute GVHD. So we say quick engraftment, yes. We have a quick engraftment, so we can form it, we can go forward with this. We confirm the data of the publications. Acute GVHD, so we say, no, reduce GVHD, this is our aim. And I think with no grade 3, 4 on 0, we reach this, but it's only outlook, it's only the first 100 days. So we have to look in this data quite more actually, because now it's getting interesting what happens after this 100 days, because now we have to see what is about CGVHD. And here some other data, so overall survival on this time point is quite high. We have got really nicely shown here. We have one patient relapse, one was a sepsis, and we have cumulative TRM was really quite low. And last but not least, to really come to the immune-competent graft, actually, was the infection. And here on the infection side, actually, we have, which is most likely common, actually, in children, is ADV infection. But we have a really low CMV infection rate, reactivation rate, and due to CD19 on a good depletion rate, 0 EBV. This is really outstanding. At all, we can say, we talk about reactivation, that means in our case, for the first 100 days, no lethal viral and fungal infection to improve immune recovery. And this actually is it's a summarization actually. We have also experience in HHV6 and BK-wise. BK-wise, we have 2 patients with hemorrhagic cystitis. At all, we still have no lethal or viral infection. And actually, in the summary on this, and for the conclusion, I have to speed up, so I'm finished. For the conclusion, actually, what I said, no lethal viral infection, We have a low incidence of acute GVHD and no treatment toxicity in TAM. So at the moment, we can say from this outlook, we fulfill and confirm what we have seen also in other hospitals, with a quick engraftment, with a reduced GVHD, reduced rate of infection, and towards an immune-competent graft. Thank you very much. So, if you have some questions, please. During the lunch, of course.
A quick question answer session if anyone has anything to ask.
The ones, 40% of the patients had adenovirus claim.
Yeah. Should I go back?
Okay. What do you do with those?
What did you do? Actually, I have asked Peter Lang for that. No, what do you mean, the treatment for them?
Yeah.
I can't say actually what they treated because...
I asked that question because, using serotherapy in our settings, we are seeing quite high symptoms of viral patients and especially adenovirus, and we definitely don't have much for adenoviruses here. Antivirals are expensive, cell therapies we don't have. So what we have done is we have changed the strategy of changing, stopping serotherapy altogether. We don't give any more ATG. At least at my center, I don't give any more ATG. So we replaced by 2 days of TBI, and that has shown a better immune reconstitution than using ATG during the serotherapy, because the initial few patients really struggled with a lot of viral reactivations. So that's what the question was. If you got access to cell therapies, you're using CTLs, you got brimcidofovir. It makes a little bit of sense to continue with that, but otherwise some of us here, we have stopped using serotherapies because the immunoglobulin is very slow when you use ATG.
Okay. On the ADV side, I can't say actually what was follow up actually on this therapy, because it's the first time I saw in the paper at the Marchand group they got really an incident on ADV. I know actually in India, Cytovir, Ganciclovir, they need to be really bring into the country. You need 10 days. It costs a lot of money. It's not easy. To have other therapy forms is quite an option. Really here, for actually, I think this is in detail a question to the physicians. Actually, you might have the possibility of EVNT. I'm sorry for that, but because certainly on EBV, I think EBV is one of the really chronic rough ones actually. It's not easy to get them so easily out. I saw also that therapies which were perfectly, that the patient after 2 years experienced the comeback on the ADV. So it's not comparable on CMV and EBV, which you really get rid of this. On ADV is a special kind and needs also a special treatment. Maybe TBI is a good solution for that.
Alright, thank you, sir. I request everyone, since we have to move forward with the show, so any other questions? So it's probably always free to answer them after the lunch. Right, sir? So I would like to thank Dr. Nicholas to actually emphasize on such wonderful clinical data results. So now moving on. I would like to introduce our next speaker for the day. She is currently working as a scientist in HLA and Immunogenetics Laboratory in the Department of Transfusion Medicine at Tata Memorial Hospital. She's completed her PhD from AIIMS New Delhi in Immunology and Immunogenetics, in which her focus was mainly on MHC and infection, with emphasis on leprosy and tuberculosis. Post which she completed her postdoctorate in studies of non-MHC genes as well as pharmacogenomics in mycobacterial infection. Currently, she is working and she is studying on evaluation of epitopes with the presence of anti-HLA antibodies in mismatched haploidentical hematopoietic stem cell transplantation. I would like to introduce Dr. Meenakshi Singh to actually come and carry forward this speech on NK cells applications in malignancies.
And we're speaking on the natural killer cells, implications in hematopoietic stem cell transplantation and not application. Dr. Rahul gave an excellent presentation on CAR T cells. So what I'm talking about is on the universal CAR cell, the natural killer cells, which responds not to the presence but to the absence of the self-HLA classical ligands. my talk, I'll give an overview of the immunobiology of natural killer cells, the KIR receptors. HSCT is the treatment of choice for patients suffering from leukemic disorders. Selection of donor is based on full match of HLA. There are alternate donors for the hepatotransplantation. However, in spite of HLA match, the overall survival rate after the HSCT is only 40 to 60%. So there are other genes which are involved in the implications, which are the non-HLA genes, the natural killer genes. Why the natural killer cells? The natural killer cells are the first innate effector cells which are reconstituted post-transplantation. Dr. Nicholas mentioned the NK cells, they come up, they go down, the levels go down, and then they form a plateau. So what happens is the natural killer cells are reconstituted first, then they perform the action of graft versus leukemic. They cause the lysis of transformed or the virus-infected cell. The levels goes down. Then are the T cells which are reconstituted, which come up. Therefore, the level of the natural killer cells plateaus down. The alloreactivity of NK cells is based on the balance of activating and inhibitory receptors. These are nothing but these receptors which are present on natural killer cells. There are two types, the activating and the inhibitory receptors. The ligands for the KIR or the natural killer cell is the HLA class 1, namely the HLA A, B and C. These KIR molecules are present in human chromosome 19q. They are coded by 9 exons. There are a total of 16 gene loci, which have 8 inhibitory, 6 activating receptors and 4 framework genes. How these KIR receptors are named? The naming of KIR is based on the structure, which is the number of the extracellular domains, the 2 domains or 3 domains, they call as KIR2D or a KIR3D, and based on the intracytoplasmic chain, the long chain or the short chain, they are named as either inhibitory receptor or as an activating receptor. Therefore, a KIR receptor with a long chain is an inhibitory and with a short chain is a activating receptor. So, how do these function? These KIR receptors have something called as the tyrosine-based inhibitory motifs, which they are activated upon encountering the ligand, which is the HLA class 1. They are phosphorylated. The phosphatases get activated, which leads to the suppression of the activation signal. The ligands for the KIR are the HLA class 1, the ABC. The KIR2DL2 binds to what is called as the HLA group C1, which is coded by asparagine acquisition 80. The KIR2DL1 binds to HLA group 2. The 3DL1 binds to the BW4. The BW4 epitope is present on 40 to 60% of the HLA-B alleles, whereas 3DL2 binds to HLA-A. The reason of showing this table of the KIR and the ligand is to understand how we screen a donor based on the HLA, the ligands and the KIR genotyping of the donor. So what happens is when a donor has a KIR2DL1 and it encounters a ligand which is C2 for the 2DL1, this will lead to a stronger inhibition of the lytic activity of the NK cell. The main function of NK cell is the lysis of transformed or a virus-infected cell. So when a K2DS2, which is an activating receptor, encounters a C1 on the graft or on the patient cell, it causes stronger activation of the lytic activity. So apart from the KIR receptors, the variability of KIR is determined by the haplotype which are present, the group A, the group B and the gene motifs. So every individual has either a group A haplotype which has more of inhibitory receptors and less of the activating receptor. On the other hand, conversely, the group B haplotypes have more of activating receptors. There are 4 motifs, 2 haplotypes for each individual and 4 motifs, the centromeric motif and the telomeric motifs. This cartoon, it depicts the function of NK cell. So now NK cell elicits a response based on the receptors which I just depicted. What happens in a normal condition is the NK cell looks at the resident healthy cell, it encounters or the receptor on the NK cell finds the ligand, which is the HLA class 1, the function, which is the lysis of the cell, does not happen. So the resident healthy cell remains in the system. However, in a transformed or a target cell, the receptor, the ligand for the receptor is not present. This does not lead to the function or the inhibition of the lytic activity of NK cell is lost. This causes the NK cell to remove the transformed or the resident leukemic cell. This is what is called as the graft versus leukemia effect. So when an NK cell encounters an autologous or a self cell which has the expression of HLA class 1, the lytic activity, the NK cell activity is not elicited. However, in a tumor cell, there is a missing self. The HLA class 1 is lost. It elicits the function which is to kill or to remove the target or the transformed cell. This mechanism or this function of natural killer cell is what is utilized in the clinic. So based on the function of NK cell, 4 models of alloreactivity of KIR have been envisaged. So one is the ligand incompatibility model. This is a model by the Perugia group wherein they looked at the high resolution typing of the donor and recipient, the HLA typing. They assume the KIR genotyping of the donor. On the other hand is the receptor and the ligand model where the HLA typing of the target cell and the KIR genotyping of the donor should be known. The missing ligand model is the HLA typing of the recipient is understood, whereas the donor typing is assumed. The other is the KIR gene model, the Stanford model, where the KIR haplotypes and the motives of both the patient and donor are taken into account on deciding the best donor for our patient. So when between a patient and a donor, when there is a KIR and the ligand match, the 2DL1 binds to the C2. There is inhibition of the lytic activity. However, when the K2DL1 does not encounter its receptor, the ligand, which is the C2, there is a GN activity which is undertaken or which happens in the graft versus host direction. This leads to the removal of residual leukemic cells. So basically, the cells which are present in the patient, they are utilized to remove the resident or the refractory leukemic stem cells. This explains the same phenomenon where the different receptors present on the graft, they encounter the leukemic cells on the host. Killing or no killing is directed. So with this understanding, we took a study which was on 33 patients which underwent the haploidentical T cell replete HSCT. They were not depleted. So we tried to use the cells which were present in the donor as well as the host to induce a leukemic effect. The median overall survival was 13 months and the median relapse-free survival was 12 months. The disease status at transplant was clinical remission for majority of patients followed by persistent disease. The mean age of donor was 34 years. Transplant with donors more than 30 years resulted in favorable outcomes. This is in converse to what is being published in the literature. They say the younger donors, they perform better when they're taken for transplantation. Majority of the donors were male gender. Majority of the transplants were performed with sibling as a donor, followed by mother, father, and a son. The HLA and genotyping was performed by the PCR-SSP method. The KIR ligands were assigned according to the algorithm which is published or which is available at the Anthony Nolan site. The KIR genotyping was done looking at the 14 activating and the inhibitory receptors. So what we observed in these 33 patients was Where there was a kill-ligand mismatch between the patient and the donor, the overall survival, the relapse-free survival was better. However, this did not reach the significant level. 5 out of 33 patients were kill-ligand matched with the donors. On the other hand, when we try to look at the patient's C ligand and the BW4 epitopes and correlated with the overall survival and relapse rate, what we observed was the patients which had the C1 ligand and there was absence of BW4 epitope. These patients had 75% overall survival and there was no relapse. So this was highly significant when we looked at the survival curves. Patients with C1 and absence of BW4, the overall survival as well as the relapse-free survival was significant compared to patients which had C1-C2 or a C2-C2. In short, what it actually means is when the patient has absence of HLA ligand, which for the receptor which is present in the donor, there is removal of residual leukemic cells. The graph does well. There is the chances of relapse are much reduced. So we were not, in this case, we were not giving any immunotherapy. We were using the graft NK cells to induce or remove the leukemic cells. Then we also try to look at the donor genotype. The donor genotype, when the donor had a telomeric B type, the AA or the B type, with patient carrying a ligand C1, the chances of the overall survival was better than a donor which had a KIT A genotype. This showed a trend but did not reach a significant level. So the data, when we analyzed, led us to form an hypothesis which we implemented in our clinic, wherein the donor is not only screened based on the HLA type, but is also screened based on the HLA ligands which are present in the patient and the KIT genotype which are present in the donor. What I want to tell the audience is that these are not the AML cases. The data which is published in literature has been shown that the effect of natural killer cells or KIT receptor, it outshines when there is a patient of AML. This was a mixed bag, the pediatric and had both the pediatric as well as the adult patients. So there were these two cases where case of AML and CML, the donor for the case 1 was brother and the donor for the case 2 was the father. It was GVH, in the GVH direction between the graft and the host, it was a 10-on-10 match only at one feed, not at the subtype level. So when you look at this HLA typing, we're looking at just the allele family or we are looking at the peptide binding region. So this was only at the allele family matching. It was matching 10 by 10 at 5 loci. However, in the HVT direction, that is between the patient and the host, it was a 5-on-10 match. So both these